All right, we're happy to have here Yaniv Assaf with us, and he's going to talk about the role of neuroimaging in redefining neuroplasticity beyond the synapse.
So thank you very much, and indeed plasticity is the topic of what I'm going to speak today, and basically this is what we want to know. So when we are talking about the brain project and brain mapping, we do want to know where every experience that we undergo is absorbed in our brain in order to map it, to understand why one person is that way and another person is another way. So this is one of the great challenges that we have. And on the very, very basic and wide definition that we can give this process is the ability of the brain to change itself functionally and structurally. We'll give an emphasis in this talk on the structural aspect as a result of cognitive experience. So we have a brain, a cognitive experience, and eventually we want to map that. What happened to the brain in terms of the structure after we acquired some kind of experience? So when you start a project on any subject, you need first to go to understand what is in the literature. And in the literature, when you open the textbooks in neuroscience and neurobiology, you find this. This is the processes. When you go to the chapter of brain plasticity, these are the processes that you find. Long-term potentiation, synaptogenesis, axonal sprouting, maybe, if we are young enough, some kind of genesis of different kinds of cells. This is what we have. This is what we have in the brain. And that's fine. 100 years of neuroscience led us to this, and this is actually the basis for some of the projects that Idan mentioned before. The thing is that in the recent 15 years, there were several works with MRI that gave another aspect for plasticity. For example, the seminal work of the Maguire group of London taxi drivers, implying that there might be some kind of long life plasticity in the hippocampus. Their hippocampus was larger due to their experience. Then the very controlled study of Dragansky from the May group in Germany, showing that a few weeks of training in juggling caused increase in the cortical thickness. So this method measures the cortical thickness, increasing the cortical thickness in relevant brain areas. And since then, there are quite a few works that show different domains that undergo significant changes in the volume of the cortex after experience. And then that could be experience a few weeks. And also in rodents, it was shown in the water maze. And now we have a gap, because these effects that we do know from conventional neurobiology, conventional neuroscience on the synapse level can hardly explain what we see with imaging. Obviously, imaging catches something else, and I want to explain why that is. So this is the synapse. This is what we know about brain plasticity. Everything we know about brain plasticity is on that level. And just to give you the analogy of what we catch with MRI, so MRI sees something, and we need to understand what is the relevance between what we see with MRI and the relevance to the synapse. So let's take a person, put a circle around it, and put it in the relative volume of a synapse as the synapse to an MRI voxel. What would be the voxel that will catch a person? So a 1 meter. So this is a voxel. Can you see the person in the voxel? Can anyone see it? It's there, believe me. So that means that in a radius of 100 kilometers, around one person, that would be the effect of one synapse on an MRI voxel. This is negligible. You can hardly even think that a synapse will affect anything that we measure on the MRI level. Obviously, MRI is not sensitive to things that happen on the synapse level. But we do get something with MRI, and MRI, believe me, doesn't lie. So we get information from MRI. It does show plasticity. It shows that something happened in structurally. In the gray matter, in the cortex, and we'll speak later on other areas of the brain. But we cannot explain it with the synapse. Yeah. So there are some open questions that you need to answer. So what is the relation between the microstructural plasticity, as we know it from conventional neurobiology, and macrostructural plasticity, as was revealed with MRI? What are the biological correlates of what we see with MRI? And also previous MRI experiments were done on a few weeks of training, and we need to know what happened on shorter timescale. Obviously, the few weeks of training caused increase in the volume of the cortex. It started somewhere, and we need to find out where. And to try to answer the question, we need to change our methodology or to get something that is more specific. So the conventional MRI, just as I showed you before, looks at the thickness of the cortex and compare it as a function of training or whatever, a function of time. But in order to get more specific at what's happening or to get an information that will be closer to things that happen on the synapse level, we need to get to the connections themselves. And just taking this definition from ourselves, Meshulam, that nothing is defined better the function of the neuron than the connections it forms. And if we were able to measure the connections, I don't need to see them. To see them, I need to take the brain out and look under the microscope. But just to measure them somehow, quantify them somehow, maybe I will be able to say something better about this process than just saying that it's happened on the synapse level or that the thickness of the cortex increased without saying what happened there. And we do have a method in MRI, so I'm not going to go into the methodology, just explain a bit what we are doing with it. So we do have a method with MRI that can see or can give us some quantitative information about the connections. So this is conventional MRI of the human brain. But with diffusion MRI, we are able to get into the brain and look, if that works, no, and look at the different connections of pathways that we have. And we can quantify them even on the long scale. This is where we use diffusion tensor imaging to extract tracts, but also on the micro scale. For example, if we look on the rodent brain, so this is a rat brain. This is a diffusion imaging of the rat brain. This is the cortex, the hippocampus, and the corpus callosum. We get a lot of information in the corpus callosum, but just putting the information from diffusion imaging on histological section of the same tissues, and this is a MAP2 staining of the brain showing the dendritic tree, the different connections between the neurons. And you can see that the information that we get from diffusion imaging is well aligned with the direction of the dendrites. So if we can quantify this information, doesn't matter if on the white matter or on the gray matter, we will get some kind of quantitative information on the connections that we have. So taking this methodology with us, we are going to look at brain images as a function of some kind of experience, both in rodents and in humans. And there are several parameters that we can extract from this methodology. The most basic of them is called the mean diffusivity, as you can see here. The most famous of them is the FA, the fractional anisotropy. It's the most widely used DTI, diffusion imaging parameter in neuroimaging, as well as structurography at the end. In this presentation, I'm going to focus on the mean diffusivity, just because it's the most robust parameter that we can extract. It's homogeneous across the brain. The acquisition of this parameter is extremely fast. Within really few minutes, 3, 4, 5 minutes, you can acquire a whole brain of this parameter. It's well defined in all types of tissue in the brain, and the interpretation is simple. The denser the tissue has, the more connections it will have, the slower the diffusion will be. So what we want to gain by using diffusion imaging to look on brain plasticity. So first, we get more specific information on microstructure. It's not just that we measure the cortical thickness, but we are diving into the connections themselves. We actually exclude the volumetric effect because in the process of the analysis, we just normalize the brain. So all of them needs to have the same structure. We just take the brains, deforms them, so they will have the same structure, and then just look on the quantity. The quantity is the microstructure, is the mean diffusivity at the end. And this is the time scale that is going to, so now we're going to see some examples on how diffusion imaging can look or can get us some information on brain plasticity. And this is a time scale that is going to be used throughout the presentation, where we'll see what will be the effect of plasticity on diffusion imaging on the week's timescales, and then we'll reduce, we'll see until when it will be sensitive enough. So the first experiment that I'm going to show you is using enriched environment in mice. In enriched environments, it's a very known paradigm, taking the mouse out of their regular cages and putting them in cages where they have, so it's larger cages, they have many toys they can play with, many places where they can hide, more significant social activity, etc. And it is known that this kind of paradigm it causes increase in learning and memory, and eventually increase in neurogenesis and plasticity as well, as was shown in previous studies. So what we have done, we've scanned these mice for a period of 2 months. The mice were placed in the enriched environment or in a regular environment for a period of 2 months, and afterwards we scanned them in the MRI. And these are the results. So what we see, and I want to spend a minute just to explain the type of presentation, because this is going to repeat throughout the presentation. So what we see is a mouse brain. It's an MRI fractional anisotropy map of diffusion imaging, although the information, the colored information relates to the mean diffusivity. And the colored area, they are not fMRI. Usually we're used to see it in fMRI. These are not fMRI. They're just areas that shows places in the brain or voxels in the brain in which the mean diffusivity, the quantitative parameter that we extract from diffusion imaging, has changed between, for example, the two groups here, the enriched environment and the regular environment. And we can see a large sections of the brain that are involved. The primary sensory cortices, some of the subcortex are involved, and in all of them, we see reduction in the mean diffusivity, meaning that the tissue become dense in that areas, in a way, increasing, if you will, increasing the connectivity or increasing the ability to transfer information between the different cells. What we do know about this paradigm, that specifically in the hippocampus, one of the regions that were affected, we do know that there is increase in synaptic activity, as was shown by previous work. But we just said in the beginning that it's very unlikely that MRI will be able to follow anything that happened on the synapse level, but obviously we do get the highest correlation between changes that happen on the mean diffusivity and things that happen on the synaptic level. Without the ability to give this better interpretation, we moved on to do another paradigm. And the paradigm that we chose, is just in order not to make a weeks of training, but a shorter time scales of days, and we choose the Morris water maze. So the Morris water maze is one of the most widely used paradigms in behavioral psychology. It is well localized in the brain. We know exactly which brain areas are affected by this paradigm: the hippocampus, cingulate cortex, entorhinal cortex, fimbria, fornix, etc. It is relatively fast training, 5 days of training. It is controllable in the sense that we can make the paradigm controlling for different aspects of the learning. So there is a motor learning in the task. It's a pool. I will show you in a minute. They need to swim. There is spatial navigation parts of. So we can control for each part of the parts of the task, and it is known to cause plasticity and enhance learning and memory. So in this task, you take rats, you put them in the pool. There's a hidden platform in the pool. And eventually, so they are trained in the pool 4 times a day for 5 days, and eventually they do know where the platform is located, just based on spatial cues that are around the room. In terms of the experimental design, we have the MRI at baseline, so we measure the same rats. It's not different groups; it's the same rats. We scan them at baseline before they did any cognitive task. Then they have the 5 days of training in the water maze, and eventually we put them back in the MRI just to see what happened. So this is, for example, the path of that one rat did at the first day of training. You can see that it doesn't know where to go. It just swims at the borders of the pool, eventually trying to explore to deeper parts. And finally, if it didn't find the platform, we just put it on the platform that it will know that it is there. But you see that from day to day, the path that it goes, it's much shorter. When we put it in the pool, it knows exactly where to go, and it finds the platform at the end. So you can see, hopefully, a short movie showing this rat on the first day. Yeah. So it's just wandering around, trying to understand why we put it in the pool. But the same rat, so we'll let her swim, but the same rat at the 5th day, it knows exactly where to go. She will just swim exactly to the platform and will find it. So good for the rat, she found it. And we can extract some parameters out of it. For example, the latency, the amount of time it took the rats to reach the platform, and we see a very nice learning curve. But now we have the MRI before and after the task, and we can ask which brain areas change their microstructure, the connection, as we measure it from diffusion imaging in this task. And we do find much focal regions that were found in the enriched environment, which is expected because this is a less dramatic task. But we do find the dentate gyrus of the hippocampus, some parts of the sensory cortex, especially in the bowel field, the amygdala, obviously, entorhinal cortex, piriform cortex, and a few other regions eventually. And in all of them, again, the mean diffusivity, our marker, our parameter, reduces after the cognitive experience. Nothing happened in the control groups. So it seems that we have a very nice marker that can map us areas that underwent structural plasticity on the microstructural level. Again, in order to understand what happened, we stand here again for the marker of synapses. So synaptophysin is one of the markers of synaptic activity, one of the most common, and we stand in the areas that we found changes, and we do see that it's probably more difficult for you to see, but it's become more reddish, meaning that you have more synaptic activities in the area. Again, giving us the link between synaptic activity and MRI changes. As we said, it's unlikely, but seems to happen. Regardless, if we can explain it or not, we have a very good marker, again, for structural plasticity. So we saw that it is good for weeks, days. What happens on the hours time scales? So the paradigm, again, we use the same water maze, but instead of doing it for 5 different days, we do it within the same day. Within the same day, it means that within 2 hours, the rats undergo 12 trials in the water maze, and they are able to learn. It's a completely different memory mechanism. Obviously, in 5 days, it's long-term memory, already consolidated memory. This is working memory, immediate memory of the rats after 2 hours of learning. And indeed, we do find changes in different brain areas, different part of the hippocampus is involved. Obviously, this is different type of memory, but again, after 2 hours, we do see reduction in the mean diffusivity in the learning group, nothing in the control group as well. Staining again for synapses shows the same pattern, 2 hours of training, structural plasticity, microstructural plasticity in the hippocampus, increased activity in synaptophysin. There must be something else causing this effect. As we said, synapses are not likely to do anything. So we try to stain also for dendrites. Dendrites are in green here. You can see this is the marker map 2, but we found anything. Dendrites did not change dramatically in this task, but synapses are. So maybe it is. Maybe we do measure with the mean diffusivity something related to the synapses, although we said it can't be. Again, weeks, days, hours is okay. What happens on the minutes time scale? Here we use the fear conditioning. The fear conditioning is I think, the paradigm that the rats learn the fastest. So you put them in this cage, and every time they hear a tone, they get an electrical shock. And it takes one electrical shock in order for them to understand that the next time that they will hear a tone, something will happen, then they will freeze. So it's very fast learning, only a few minutes of training at the end. And we do find significant areas, again, of reduced mean diffusivity in the cingulated cortex, cingulum, auditory, sensory cortex, amygdala, obviously, and hippocampus. All are well in line with this paradigm. We do know that these areas should be affected. And in all of them, the mean diffusivity reduces. So few minutes of training, this is it, reduction of mean diffusivity that we can extract. And this is very nice. We have a very nice method that can help us map structural plasticity. Even if we don't understand the biology behind it, we can use it in neuroscience in order to map this region and to look at different cognitive domains. And things that can happen on the mouse level or the rat level, obviously we are all mammals, can happen on the human level. And I have a good friend, Ruti Novik, with whom we wrote together a book for kids, that when I told her about this study, she said, how could it be? The mouse brain works as the human brain? And I told her, yes, neurons of the mouse brain are the same as in the human brain. And then she looked at me and said, maybe in your brain. My brain is something else. So I'm sorry, Ruti, our brains are the same. The rats and the human brains are built of the same building blocks eventually. But you need to choose the paradigm. So if we had a water maze for the rats.
What's a building block?
A cell, a neuron or
How do you know?
Let's get it to the end, all right? I'm getting to that, believe me.
You make statements that are very important. You just said, and there are lots of students here, that the rat in the human brain, or the mouse in the human brain, or the salamander in the human brain, we can continue until one, they have the same neurons. Nobody knows that.
All right, I accept that. I accept that. The thing is that if you want to do a water maze in humans, especially we are getting to the summer, getting to a pool could be not a spatial navigation task, but rather a survival task. So we need to come up with something different. And the thing is that the students said, okay, if you're working on humans, we can use something that is more relevant. And they suggested to use the Need for Speed. The Need for Speed is a computer game, a car racing game. How many of you play this game? No, don't be shy. It's good. Believe me. Don't be shy. Come on. So in this game, we save no money. We took a Porsche. We gave our volunteers a Porsche and sent them to the streets of Monte Carlo, as you can see here. Before we gave them to play the game, we put them in the MRI. And then we gave them to play 16 times the games, split it into 4 rounds. And then we put them in the MRI again. The difference was 90 minutes before the first and the last scan, before the first and the second scan. In between, we gave them, after each round, we gave them a blank sheet of paper and asked them to draw the track. And you can see they didn't see the track, never. They just drove the car. That the map of the track was formed in their brain, eventually. Over here. So there is a component, so this is a very complicated task. There are many types of memory involved here, but obviously spatial navigation or training is deeply involved here because they do have a map formed in their brains. And in terms of learning, you see that the time it takes them to complete the track reduces significantly as they play it again and again and again. What we found that there is a reduction in this mean diffusivity parameter, which we can say, can't say much about him, but we can say that the reduction in the mean diffusivity is, as we saw in mice and rats, is similar in humans as well, regardless if their neurons are the same or not. But we see reduction in the mean diffusivity, nothing in the control group, without getting into that. We do see some other brain areas that are, so this is the hippocampus, we have the parahippocampus involved, the insular cortex as well in this task. The parahippocampus is of particular interest because we saw that the improvement rate in the task was in correlation with the diffusivity changes there. There was also some gender effect. I don't have time to go into it, but just think of the task and think of the probable reaction of each of the genders to the task. I can tell you that one of the genders had more significant emotional reactions toward the task and was found to have even changes in the caudate head, part of the reward system in the brain, if you say. Again, we don't understand the biology behind it, but we do have a very nice method that can help us now to map different brain areas after some cognitive experience, and it can be done on the very short time scale. So here it was 90 minutes of car training, but we also had an experiment when we trained the subject the names of flowers whom they didn't know, so they had to make a link between the flowers and the new name, and they had to repeat it for a period of 1 hour, leading to changes in some relevant brain areas, the IFG, the STG, hippocampus as well. Also an experiment of motor sequence learning. So in this task, the subject had to learn the first 15 notes. So they are not piano learners, so they had to play the first 15 notes of Fou-Release. You know Fou-Release? Ta-na-na-na-na-na-na-na. Yeah. So they had to play it. I don't think the student that is doing this experiment will be able to hear this melody once again in his life. But he did found, again, reduction in the mean diffusivity in the relevant brain areas, premotor areas, and cerebellum. So it's a nice method. It's a nice method that can help us map structural plasticity. But I'm in the Department of Neurobiology, and my colleagues in the department, some of them, are less impressed by the nice images of the MRI, and they're always asking me, okay, that's nice. That's really nice. What is the mechanism? What is the mechanism behind it? So what's causing that? You said in the beginning that synapses are unlikely to cause this effect. So who should we blame in this kind of result? So to do that, we need to dive into the composition of the brain. And just taking the numbers out, we'll see that the most of the composition of the brain is glial cells. 70 to 80% of the cells are glial cells. We can argue about this number. Some would say it's higher. It can be 90% in some regions. Some will say it is slower. It's smaller. It can be 50%. But obviously, this is a significant chunk of the volume of the brain, both in humans and rodents. A big chunk of the volume. And if we are measuring it with MRI and we get a signal for the water, obviously the water of the glial cell will contribute as much a signal or even larger signal, stronger signal than the signal that comes from the neurons, or the signal, if any, that comes from the synapses. So we need to understand what is going on with these glial cells, and if they are affected by plasticity, maybe they can explain what is going on with the experiment that we see. So a few words about the astrocyte. So the astrocyte, if we go by Google, when you write neuron or synapses, you get about 17 million hits. Synapses, less than 1 million. Obviously, much less studied. Astrocyte, much less studied cell of the brain. 50 years ago, it was thought to be only supporting glue, and we do need a lot of them just to put the neurons together. But 15 years ago, we did understand that they regulate blood flow. Some of these experiments were understood following fMRI. We do know that Einstein's brain had more astrocytes. It didn't have more neurons, but probably had more astrocytes in his brain. In the recent 2 years, there are more evidences that astrocytes, some say drive neuronal activity, some say that they have active role in cognition and memory, in a sense. So this is not a negligible part of the human brain. Obviously, the synapses are extremely important, but when we look at the brain, we need to look at it as a unit. And it's not something that we need to say today. It was said already more than 100 years ago by Santiago Ramon y Cajal, that said the prejudice that the relation between neuroglial fibers and neuronal cells is similar to the relation between connective tissue and muscles or gland cells, that is a passive weft for merely feeling and support, constitute the main obstacle that the researcher needs to remove in order to get a rational concept about the activity of the neuroglia, or in that sense, the activity of the brain. If we look only on one part of the package, we might not understand what it is doing, or we might get a misleading understanding of how this process works. We do know from other studies, not related to MRI, that focused on glia. So glia was and especially astrocytes were studied a lot in relation to brain injury. And we know that in brain injury, astrocytes become reactive. They become swollen, as you can see here. The processes become more pointy, much more processes eventually. And when you look at that image, you can think of, well, if you have more processes now and the cells are swollen, maybe this will affect the diffusivity that we measure. Obviously, the synapses is far too small to affect anything related to diffusion of water molecules, but this might be the fact. And a few years ago, Schumer showed that using two-photon microscopy that you can do an imaging of neurons and astrocytes together, and you can do induce activity to the neurons that are shown in green, and then you can see a large area of a large network of astrocytes that work together with the neurons. So let me play the movie. It will be better than the words. So, yeah, it will be better if you could see it. So what you could see are neurons that are working, and behind it a network of astrocytes that are working with it. And if when they are working, or they are getting to the level of plasticity, the neurons, these changes happen on the glial level, then maybe this is the thing that I catch with the MRI. So maybe this will work. These are confocal sections showing neurons in red and astrocytes in green from our rats. So this is rat that did not perform the water maze. You can see the astrocytes here in green. This is nice. But look what happened to the astrocytes after the water maze. This is a huge structural change, and this is a day after the water maze. It's not just immediately after the maze. So the changes, the structural changes these astrocytes went, it's persistent. It stays for few hours, maybe few days after the effect. And if this will not cause changes in diffusivity, the marker that we measure with MRI, I don't know what will. Obviously, this is a very strong a very strong candidate to be the cause of the effects. So just have a very, very short mid-summary, which will be obviously the ultimate summary. So what I try to show you is the diffusion MRI changes are co-localized by increased synaptic activity. But the lead candidate to cause these changes is the astrocyte. And significant remodeling of the astrocyte morphometry, swelling and processes formation seems to occur following LTP. I didn't show you, but there seems to be a relation with the amount of LTP to the amount of astrocyte remodeling that happens. And in many senses, so the basic rules that we know in neuroscience, that things happen on the synaptic level, they are true. But we need to integrate more information to it from different type cells. So keep calm and break all the rules, and if you break them completely. So I'm not going to go into that, but we do know that it's not only the cortex that undergoes changes, but also the white matter. And there are more evidences that our white matter is not a stable system. It's just constantly changing depending on what it needs to do and how much of information it needs to pass. So I'm not going to go into it, but believe me, it's happening. Yeah, just getting to the summary. So this is the real summary. So the mean diffusivity is the excellent parameter that we have just to map brain plasticity, all the other things we said. But to get a better interpretation or biological interpretation of that with MRI, we need a more specific method that we do have under our hands that are able to look at neurite density, axon diameters, and cortical layers assembly as well as other things. So I will just stop by saying that we are in the level that neuroimaging is going to have a very significant role in redefining the neurobiology of plasticity. Obviously, not everything that happens in plasticity happens on the synapse level. There are other contributors as well. It might be difficult with conventional neurobiology method to look at all of them together. It can be tedious to have 10 different markers for the same area. But with imaging, you use the same marker for all of them, so it's not specific, but it sees everything. And it can help us to redefine which brain areas or the connections between the brain areas or how the brain as one integral system undergo this process. So to thank my collaborators, and finally, thank you. Yes.
I was wondering whether you looked at the resolution of the effect. So how long does it take for, is there a reversal actually?
Yeah, there is. There is a reversal. The timescales are, well, it seems that they are related to the strength of the experience. So the stronger the experience was, the longer the experience goes, then the longer it will take to be reduced. But it sustains quite a while. And we are working on it. The one thing that I wanted to mention, Uri, I just looked at Uri and I remembered that we do have a new MRI system in the university that arrived last Sunday. And obviously these are one of the experiments that we are going to work and also look at time effects. It just necessitates a lot of subjects and subject coming again and again to the MRI scanner. So we just need a scanner that will be, that we can use regularly. So yeah. More questions? Yeah, maybe just to say a note for Yadin about this question. Yeah, just a note. You asked me a question in the middle, and I do need to answer. Yeah, we don't know. We don't know. But under the microscope, a neuron of the human brain and the neuron that you take from other animals, they look the same. And they do have similar systems. So they work through synapses. You do have glial cells around them. And plasticity seems to affect them in the same manner. So in that term, they are comparative in a sense. Yeah, I know that you know that, yeah.
If you fly in a jumbo jet and you look down and you see people walking on the street, they look the same.
Yeah. Yeah, I know that. Yeah.
What I wanted to say is that we should be careful in statement because the young generation is here.
Certainly. I agree with that.
But they have to have something to break.
Hi.
So give them an opportunity. Thank you very much for all the speakers.